Hi, so this lesson is on transpiration, how we can measure it using a potometer, the factors that can affect the rate of transpiration, and how plants cope with different levels of water availability. So it's all based around your iBook, around this page here. So we defined transpiration yesterday as the pulling of water and the dissolved minerals up through the plant. We have the cohesive forces between the water molecules, and we've also got the adhesive forces, which the lignin in the xylem vessels and the water as well. Right, so the piece of apparatus is called a potometer, and this is basically a setup where you take a cut piece of plant with the leaves still on it, and you have a very narrow piece of tube. You have a reservoir here, and if you have a small bubble of air in there, then as the water evaporates from the plant, then the bubble of air will move along, and you can use a scale to measure it there. You can also have like a syringe set up here, which after you've got your first set of results, you could plunge down the syringe and you could push the bubble back to start, so you could get another set of results. I'd recommend that you watch a video on setting up a potometer. Just do a search like so, because it is a specified practical, and it has to be set up in a special way. For instance, it has to be set up underwater so that there's no air locks within it, and all the joints have to be sealed with Vaseline to make sure that there's no leaks going on there. So that's how you measure the rate of transpiration. Sorry, what you're actually measuring is the rate of water uptake, which is a combination of the water transpiring from the leaves, but remember also that some of the water is going to be used by the leaves as a raw material for photosynthesis. Some of it might just be taken in by the cells to make them more turgid, and of course the cells could also be losing water by osmosis as well. So it's not the same as transpiration, what you measure with a potometer, but it should be correlated to it, so a close approximation of it. So that's the first objective, how you do it. As I said, do watch the video, because you need to, as I said, it's a specified practical there. Right, so the rate of transpiration, the factors affecting that, it's fairly obvious. Just think of your washing, drying on a line for the majority of them. So obviously, if we have higher temperatures, the water has more kinetic energy, and we're going to have a higher rate of evaporation from the spongy mesophyll, so we'll get a greater rate of transpiration. Humidity, so on a kind of humid day, you have lots and lots of water vapour in the air already, so you're going to have a slow concentration gradient between the air inside the leaf and the air outside the leaf. So on a humid day, you've got that low concentration gradient, so you're going to reduce the transpiration rate. On a windy day, then any humid air that is accumulated just outside the leaf will be blown away, and this will maintain the concentration gradient to increase the rate. Right, light intensity is obviously what we covered with stomata. So when the plant is photosynthesizing, the stomata need to be open, and they close at night to reduce water loss. Okay, so that's the second part of the objective. We then come to the third part, which is looking at how plants are adapted to different amounts of water. So here we've got classic water lily, and if we look inside the water lily leaf, then you can see we've got the palisade mesophyll here. The spongy mesophyll there has packed full of air spaces to make sure that the leaf floats. And the other obvious adaptation is that the stomata are on the top, because obviously they need to get the carbon dioxide from the air above, not the water beneath. You'll also notice that there is very little xylem within the water lily leaf, because of course it doesn't need to transport water to its cells. The cells are sitting in it, and it doesn't need this level of support from the xylem, the tough xylem, the lignified xylem, because the water is providing support for it as well. So there's another hydrophyte. This is hemlock living in the water. Right, the plant things that you will tend to think of as our standard plants are what we call mesophytes, and these have various ways of surviving the winter is the main thing that you need to think about. So we have things like orchids, which are growing as a bulb, so it will survive the winter underground and live off its food stores of starch. Down there you've got things like potatoes, which obviously potato tubers again surviving under the ground. You have annual plants which produce seeds, and the seeds overwinter and then they germinate the next year. We have trees which drop their leaves after they've recycled the chlorophyll, because they would be useless for photosynthesis when it's too cold and not enough light. So those are all ways of coping with the winter. As you can see, about to lose its leaves here. We also go to the other extreme, and we have what are called xerophytes. Any geographers amongst you might recognise this is marram grass, which grows under very difficult conditions of sand dunes, obviously high levels of wind and very low water availability because of how well draining the sand is. If you look at a leaf of the marram grass, then first thing that you should notice is that instead of being a flat blade, then it has rolled over. This means that all of the stomata are sunk away in these pits. Down at the base we've got hairs to reduce the air movement again, so any wind which is blowing past is not going to get out the really humid air in here, so it's very well adapted to reduce water loss. Also has a very, very thick cuticle as well. You've got just a close-up on the hairs, and these reduce the water loss inside, and there is the stomata sunk right down at the base of the pits there. So that's how plants have the different adaptations to water, so you've got the detail that you've gone in there. So what I'd like to do is the tasks here. So a labour diagram of the potometer, hydrophytes, mesophytes, xerophytes. If you just capture the screenshots, what I was just showing you back here of the leaf, you can annotate that. You can do the same for the annotation of the Ammophila as well. Okay, so that's what I'd like you to do for next lesson, please.